Okay. The next question is a little technical. So the next question asks, what are your opinions on violin plots and heat maps? To me, violin plots display the same information as box plot. And when heat maps are large, let's say we have greater than 30 genes and 30 samples, I find they're overloading with information and never really telling a good story on their own. So what are your thoughts on this?
Oh yeah, Violin plot and box plots are which are the most frequently actually used the plots actually in in the papers. I think Violin plot adds actually a bit more information about the distribution of the data, like because you see the width of the violin, and then that actually tells you where are those data points actually condensed. So it's still very useful to give you an extra some extra information. And for Boxplot, it only tells you the quartiles, and even very like different distribution data points. It can also have the same actually quartiles.
Yeah.
So it's good to have some more information. And in terms of a heat map, yes, I agree. Like, so when you have a lot of genes, like, or a lot of rows or columns, it's really hard to see it. But I think it's really good to see patterns. So if you cluster the heat map like by row and by column, then you will find those clusters, and that's actually very good for hypothesis generation. So you can zoom in that cluster and find what genes are there, and you can even do pathway analysis for those subsets of genes and see what pathways are activated. But I agree, like if you have smaller numbers, then you can just like put the gene name there, then it's very nice to see differences between, for example, treatment versus control sample. Yeah.
No, that makes sense. And I also feel that every visualization has a different purpose. So if you're just looking at, like, let's say 10,000 genes as like an exploratory start point, then I think heat map is not the right visualization to look at that. Perhaps you need to think about ways to further prioritize or narrow down your targets and perhaps visualize. So I think with visualization, it's also important to understand what visualization to use to effectively show the trends or patterns or visualize the trends in your data.
Right.
Okay. So moving on to the next question. So the next question is intermediate materials to help understand statistical models. So I'm currently watching, so this person is currently watching my DE-seq video where I explain negative binomial and how the use of that statistical model was arrived at. I would love to know more about how models work and how to select them. A beginner-friendly material will do too. So I remember when we were discussing this question, we talked about it's important to understand the statistical aspects conceptually, but you don't always have to go into the nitty-gritty of it.
Yeah, exactly.
Yeah, sorry, didn't mean to.
No, exactly. I mean, it can be really complicated. DSIC too, like statistical model, can be really complicated, and not everyone is Michael, the author of DSIC too. So you don't have to actually under every small details of the model, but at a higher level, you understand what the model is doing, and it's doing what you want to. Then, of course, you don't blindly apply the model, but for a tool that is so often and so commonly used, you can just use it and then try to interpret the data. I think that's what a computational biologist the job is. Just interpret the data is more important. I think you leave that heavy work to statisticians. Yeah.
That makes sense. And just wanted to say, like, this totally agree with what you said. But if there are any other statistical concepts, like basic statistical concepts, I'll always go to this YouTube channel called StatQuest.
Yeah.
That's a very famous statistical channel, and I think he does an amazing job explaining the concepts in a very intuitive way. So that's a very beginner friendly channel, and he explains all of the statistics. I think he's covered like a lot of statistical concepts.
Yeah. That's Josh Starmer. Like, let's
Yes.
have a shout out to him. And he also recently have a lot of deep learning videos. So it's also machine learning videos. So it's very good to have a high level understand how the model works, what's the advantage versus disadvantage of different models, or what kind of data type is suitable for this type of machine learning model. And I watch all those like deep learning videos all the time. Yeah.
I always go back to the machine learning concepts for like the Josh Tammer videos, because he explains the concept like so intuitively. Like whenever I need like a refresher on, okay, can we do this? Can we do that? Like just go back to his video, and he does the great job, and then I can just go ahead because I now understand the conceptual aspect of it. Then I just have to figure out the application.
Yeah. One of the earliest days, and the video that I watch is how PCA works, principal component analysis. Watch that one if you don't.
We highly recommend that. Okay, so moving on to the next question. So next question, we have grouped two questions together. So the first question is, in the context of using whole exome sequencing and whole genome sequencing tests to identify germline rare diseases, at what stage can the FASTQ data be considered failed or unqualified for variant calling? And I think we can answer this question first, and then we can go on to the next part of the next question, which is the next part of this. So I think whenever the first step we do is FastQC to check the quality of reads. And I think that is the first stage can tell you whether your data is fit to be processed further or not. What are your thoughts on that?
Yeah, of course. Like look at the quality. If the reads quality is very low, and FastQC will give you all this report and warnings, and you know whether the run is successful or not. But again, I want to emphasize that don't just blindly actually apply variant calling. Also do a little bit more actually quality control of the data. Like also, do you see any like swaps of sample swap? Because sometimes people label them just like wrong, and then you need to be able to spot those weirdness of the data. For example, if it's a female sample, then you see a variant on Y chromosome, then there's something wrong.
Yeah,
So
there's something wrong. Yeah.
yeah.
So that's going to be the next part of the question. So the next question is, how can one detect sample mix-up or cross-contamination in genomic DNA, specifically in FASTQ, BAM, or VCF files? And that's a very good point, that this is a basic sanity check. So we should have sanity checks at every point whenever we are running any omics pipeline, to make sure that, just as you said, if we have a female sample, we should not expect to see any chromosome Y. And again, there are a lot of tools that we discussed that can help to deconvolute or help to like segregate or detect the sample contamination.
Right.
One of them were, I have used FastQ and BBMap. These are again some tools that we use, and we will be adding the link to the tools that we're talking about in the description section.
Yeah.
But these are some of the tools that help you to identify if there's contamination in your BAM files, or since I deal with a lot of xenograph data, so we have tumors which are inoculated in mice and we extract them. There are a lot of mice reads and human reads. So we need to deconvolute them when we are performing any downstream analysis on that data.
I think there's other one. Is a sum layer?
Sum layer. I think we discussed about that last time.
Yeah. We'll put the link in the description of the video. Yeah.
So there are a lot of tools available, I think, and I'm sure there must be a lot of newer tools as well with improved efficiency to detect contamination and help resolve the sample mix-up. So some of the links we will be adding in the description section below. Okay, so the next question is, what is the difference between using pipelines such as Nextflow or snakemake and writing a normal R code for a workflow?
Okay, so I personally use snakemake quite a lot, so I can.
Yeah, me too.
Okay.
I haven't used Nextflow.
Yeah, so if you don't know, snakemake is just a workflow language. It's an extension of Python, and if you know Python, so you can easily just write snakemake workflows. So one of the benefits of workflow snakemake compared to, for example, just your bash scripts or R scripts, is that you can, especially when you have a lot of samples, especially like you want to process them all in the same way consistently, and snakemake is very good at that. So it will also track. So if you have multiple steps, you will track, okay, which step it failed. And when you rerun the whole process, it will only rerun the failed ones rather than just run everything. So you're wasting the computing resource. So, I mean, you can use, a bash script to write a workflow, or you can use R scripts. But again, when you want to actually run a lot of samples, snakemake will have a lot of benefits because it will track all those metadata. Yeah.
Yeah, no, those definitely I agree with that. However, I will point out some of the cons of snakemake is that snakemake has a learning curve. So when I was writing it for the first time, it took me a longer time to figure out and write the whole pipeline. And it only makes sense, as you rightly said, that if you have multiple samples, or maybe if you use that pipeline over and over again on newer samples, and it makes sense to build a snakemake pipeline. But if you're doing an exploratory analysis, or if you're doing just a one-time analysis, then the amount of time invested in building a snakemake pipeline is not worth it. So it's not really for exploratory or experimental analysis.
Yeah, exactly. So unless you're doing like high throughput or like sequence in your lab, so you when I was in MD Anderson, our lab actually generate thousands of actually data sets for chip sequencing. So that's why I have to actually make a snakemake pipeline to process all those data consistently. So, but if it's just one-off like analysis, you don't have to, and you will spend more time just to learn snakemake.
Yeah, no, that definitely makes sense. So yeah, these are the pros and cons. So one has to make a choice wisely if they want to use snakemake, and if that suits their project requirements.
Yeah. Nextflow is a different workflow language, and it's gaining popularities as well because it's very scalable, and but it's Groovy based. So although you don't need to know Groovy, it's more like a Java like language, but you can still use it. Just for me, like because I kind of know Python, so I use snakemake, but many people are using Nextflow as well. Yeah.
Yeah, I've heard a lot of people, even at my workplace, using snakemake. I haven't heard much of the Nextflow, but that could be a better version, but haven't really tried it. But and the next question in this on these lines are how to build flexible and robust code. For example, if I want to use one of my script for another analysis. So in addition to snakemake, just as you said, is useful for if you have to rerun again and again. But another couple of things that we discussed was we can have functions. So if you don't want to build a snakemake pipeline, if you're just using a specific block of code, you can just make it into a function and call that function, or source that function and just run. Or maybe if you are using that script so many times, and maybe think about a package creation, like create a package with that those functions or those scripts.
Yeah.
So yeah, I think those are some of the ways we could reuse the script for another analysis.
Yeah. So you will actually realize when you need to make it as a function. So if you are copy paste the same code multiple times, then you know, okay, it's time to wrap them into a script. Whether it's a bash script or it's a R script or Python script, so you make them more like a function or a module. So you can put them in a specific folder. Then when you do the analysis, you can just source the script from that folder, and then you are using the same function all the time. Yeah.
And the same goes for the package. Like if you're using the same set of scripts multiple times, then it makes sense to convert that into a package instead of sourcing those scripts again and again, like individually in your projects. Okay, great. So the next question is, there are two questions which we club together. The next set of question is, how does one go about demonstrating that they have skills needed to transition into bioinformatics? For instance, if someone is coming from a more traditional statistical background or social sciences background, hence already have knowledge of R and Python, but is missing skills like RNA-Seq, what is the best way to demonstrate that they have filled that knowledge gap? I know the general advice is to do a project, but I'd like to know what are the ingredients for a successful portfolio project. And the second is on the similar lines, what projects can I pick to start building a genomics portfolio, possibly leading to a multi-omics one? And I think this the answer for these questions are from our previous questions, bits and pieces that just as you said, reproduce analysis from the paper, get used to different omics types, and also understanding what each omics type, like the omics data set, what information that provides us, what are the different kinds of tools. And I think this led us to us discussing previously that especially when we are looking to hire people, I think we do not always see for the 100% match for the skill, but rather how well a person can adapt to different technologies, tools, and data types.
Yes. I think whether you are willing to learn is a very critical component, which actually I I emphasize a lot when I am hiring people. So if is this person willing to learn new stuff? And for example, for RNA sequencing data, like you were asking, like how you gain this experience, we talked about this. Okay, just go and grab public available data sets from Geo. Okay, you get raw FASTQ files, then you write down all the steps of how you convert the FASTQ to to a BEN file, then how you quantify them into discount matrix. Then you go to this downstream analysis, like differential gene expression and pathway analysis. And by the way, all those, if you want to build a good profile, put all your projects on GitHub. So that's actually the first thing I look when I hire. So whether I see your GitHub link in your CV, then I will go to your GitHub and see how well the codes are, and also how well organized the projects are, whether you have good documentations or not. So things like that.
Yeah, those are some of the ways where we can gauge how much you understand not only writing the scripts but also understand the importance of version control. How much do you use those kinds of version control methods? And again, I feel when we are reproducing, let's say it's a really old paper and you're reproducing, there might be that they might have used something, an older version of a package, or a package which was used at that time, but there are different packages. So might as, I think that's an opportunity for the candidate to display that this person has researched, understood what are the tools available for these type of analysis today, and they have used those kind of tools and shown probably if there is a difference or improved efficiency or accuracy. I think those would be the fantastic ways to demonstrate your understanding of not only the multi-omics data but also the tools, and also your understanding of these data types.
Yes. I think reading papers, definitely, then you get some sense, okay, how people actually analyze data, what tools they use. So the other way actually to keep yourself on top of the trend is to be on social media, on Twitter or LinkedIn, follow people who is doing bioinformatics, who are bioinformatics professors, and also people like me and Kushbu. So we will share the newest tools. Then you're kind of aware, okay, what kind of tools I can use for specific questions that I have in hand. Because most of the time, the question that you have or the problems you have, other people have actually have tried.
Yeah, they already have it. And I think I just, this is a completely unrelated, but I see a lot of people reaching out to me saying they have installation issues, they cannot install a particular tool. And you and I understand that it's very hard to recreate someone else's system and reproduce the error. So we usually, most of the times, we cannot help with those errors. But the best way to find out is to go onto their GitHub and issues for that package or that tool, and you see that there are hundreds of other people having the same error.
Yes.
And there are hundreds of suggestions in there where you can just go and try each one of them out. Or maybe you can just post your problem if it's a unique problem, and the package creator or maintainers will reach out to you and will help you troubleshoot that. So yeah, reaching out to the community, I think, is the best way to troubleshoot your errors. And and just to say that I learned so much from your post, all the tools that you post, I learned so much of them. I get to know about so many new tools which I did not know. So yeah, learning from each other is the best way to keep yourself updated in this field.
Yeah, exactly. So if you have an error message from your installation process, just copy that and Google it, or better now you have ChatGPT, just ask ChatGPT, but make sure you give enough context and you will get some answer from from AI as well.
Yes. So the next question, so we have exhausted all our user questions. So next question is from me.
Yeah.
And the next question is, in the integration for single cell RNA-seq data, and we did speak about this the last time, I wanted to get your opinion on what justifies the need for integration. Do we always need to do integration? How can we determine if it's appropriate to integrate or to not integrate?
Okay.
And I think you did give a talk recently, and you did give me a link to that talk. So I will be adding that link as well in the description section. So for anyone who is trying to understand integration and trying to decide whether it just whether my problem or my data set justifies integration, then we highly recommend you to watch that talk.
Yeah. So you probably will watch a lot more details in the talk, but I would just want to emphasize that integration really is trying to actually make all the samples that kind of mix together, and also to identify the cell types in a consistent way, so you can do analysis within the same cell type across different conditions or across different samples. And really, it just provide a unified way to annotate the cells. But eventually, I mean, if you do differential gene expression, you still use the raw actually counts, which is the raw data, is not after transformation. So, so there's always like, I think there's a risk always you will remove some of the biological differences when you try all those different integration methods. Just be careful like when you use them.
Yeah, that's always the thought in my mind, that if I'm integrating, whether I'm just losing out on any biological signal is what my fear is. But yeah, I think we are getting, so recently I got to know that there are improved methods which are so Sirat recently released something called as layers. So instead of having each object merging them and integrating, we can have all of that data into different layers. Sirat now allows you to, so first they just had like two methods, the reciprocal PCA and the CCA. Now they have added other methods like harmony, and there are several other methods that you can use, and you can compare across these methods on how the integration performs across these methods. And you can have all of that data in like just one object instead of having like different objects. So it's interesting. I think this field is advancing, and we are going to see a lot of improved advanced techniques to integrate such data types. And the last question is from both of us, and this is a very important question as to how to ask questions. And there's a video that you linked to me, and I think we will be adding that link as well in the description section. But after receiving these questions, we had a discussion about how important it is to ask the right question. And there's a way to asking questions to get the most relevant answers.
Yes. So you have to remember that. So if you are asking other people to help you, you need to make other people's life easier. So when you ask questions, which means you need to provide enough context. So what are you trying to do? What have you tried? What didn't work? And why you think it doesn't work? And then it's even better you can provide some of the reproducible scripts, and some other people can run it. So I think if you are not willing to put effort in drafting a good question, no one will be willing to answer your question either. So just try to understand that.
No, I totally agree. So this is a very relevant point, that a lot of times people don't put in efforts to even explain the entire context, because you or I have never seen or dealt with that data or your analysis. This is the first time we are thinking about it. And if you do not provide that kind of context, it's very hard for us to understand how can we help you. Like we want to help, but you have to provide us, as Nami said, with all the relevant information, including the things you tried. Because this is the thing that I get a lot of times, that okay, this does not work and I want to do this, can you help me with that? Yes, I can help you with that, but what did you do to help yourself?
Exactly.
If you can list down the steps that you performed that did not work, then that gives me a starting point to help you in that direction. But if you just ask me to help you, then it's a little vague request, like how can I start with that? So
yes, exactly. So it's not only about like asking computational biology questions. It's about asking any questions. So if you want to other people to help you, make sure you put some effort in drafting the questions. Yeah.
Exactly. And yeah, I think those were the questions that we received. And yeah, this was a really interesting and fun conversation. And I think we exchanged a lot of useful ideas, which will be helpful to our viewers. And if we get more questions, then perhaps we can meet again and do this again.
Yes, great. Khushbu, it's really nice to have this conversation. Thanks for the invitation. I fully enjoy it, and probably we'll meet again next time.
Yes, we will. And I'm really grateful that you agreed to do this, and I get to learn so much from you. So thank you so much for taking your time. Thank you.
Same here. Like I learned a lot from you too.